Hi, let me introduce another speaker. This is Dr. Petr Kharchenko from Harvard Medical School, and his presentation is dedicated to transcriptional impact of disease at the single cell level.
Okay. Yeah, so you probably heard this introduction already, but I'm going to talk about single cell analysis and analysis of disease using single cell data. So in general, single cell analysis have taken off over the past few years, and a lot of the efforts are really dedicated to sort of mapping out the base organization of the tissues. The Human Cell Atlas is sort of a landmark project in this regard, but also other initiatives to basically figure out the composition and the states of the cells that exist in the system. But the other sort of very prolific and probably insightful use of single cell techniques is in comparative setting. So in this case, we're trying to, let's say, compare a disease state with healthy state or treatment with control. And again, there are several consortia efforts that are sort of going along these lines. For instance, Human Tumor Atlas Network or Lifetime Initiative in Europe. And for this talk, I'll mostly focus on this comparative setting where we're trying to analyze disease and how disease changes the tissue. I'll, for the most part, use an example of our study from bone marrow metastasis. This is a prostate origin metastasis. So in castration-resistant prostate cancer, metastasis to the bone is nearly a universal feature that occurs in 70 to 90% of the patients. Unfortunately, there's no effective therapies at this point. And recent studies of immune checkpoint therapies have shown limited effect. It's also poorly studied tissue or samples because the material is pretty hard to get. We teamed up with surgeons to take advantage of rare spinal decompression surgeries to get fresh samples and dissociate them and perform single cell analysis on them. So here's briefly study design. So we take from each surgical case, we take 3 different fractions of the solid tumor itself, the bone marrow, liquid biopsy of the bone marrow immediately next to the site of metastasis, we call it involved bone marrow, as well as liquid biopsy further away, called distal bone marrow. As a control, we have bone marrow from patients undergoing hip replacement surgery. So they also have inflammation, but it's not cancerous in nature. And then we go through single cell analysis, and I'll try to walk you all the way to sort of validation experiments that we've done. The first thing to note, so here you have each point, let's say, sorry, each square is a different sample that we take, and each point is a cell in that sample. We have different classes of samples that we take. And the main thing I wanted to say is that, you know, in order to analyze these types of collections, you need to figure out what kinds of cell types. So, you know, this cluster may, for instance, represent T cells, and such T cells, analogous T cell states exist in other samples, but it's not actually trivial to recognize the correspondence. And what we want to do first, before any sort of comparative analysis or expression difference analysis, is find a way to jointly track individual cell types throughout the entire collection. So if you had, let's say, a T cell population here and a T cell population here, they would actually be labeled in the same way, and you could compare them, let's say, between conditions. If you simply align data sets, you know, put matrices together, they will not align very well because each individual has its own expression pattern, its own differences, and also there are technical differences in preparation of each sample that end up putting them in a slightly different state. So the first thing that's needed is correction during alignment that allows you to establish sort of correspondence between cells. And in this case, you know, we've designed a method for doing data set alignment based on sort of multiple reflections of different samples. Essentially, we try to find, you know, best correlated cells in other sample. And since we have many samples, there are many pairwise combinations that can be done. This forms this type of a joint graph where all the cells from all the samples participate, and we can use that graph to, well, to visualize the data, but also determine these types of joint clusters that essentially persistently track individual cell types throughout the collection, and also propagate information between the samples. So this is a published work, and there are actually quite a few methods for data set alignment these days, Syrah, Harmony, and so on. All of them have their own strengths and weaknesses, and it's still work in progress, to be honest. But one way or the other, if we have this collection, and this is our bone metastasis collection, now we have sort of a joint annotation. So, all the cells, let's say T cells from different samples or NK cells, actually aligned onto this position. Now, once you have that, you could start doing comparisons. For instance, here, I'm simply plotting these cells in different colors, and you have the controls, the benign samples, and all the cancer fractions are plotted in gray. And what you can see immediately is that, for instance, B cells are much more abundant in the controls than they are in cancer samples. For some reason, the generation of B cells is actually inhibited, and the amount of mature B cells in the bone marrow is decreased significantly. So this purely visual comparison, of course, you could put this on a statistical basis. So here we have a fraction of total cells in different samples. Every point is a sample, and we're simply looking for every cell type and what fraction did it account for in every sample. And here, for instance, these B cells mature, and you could see that their fraction was much higher in the blue, in the benign samples, and the controls compared to all the cancer samples. Now, it's a very simple analysis, but even this analysis will give you some statistically significant results in many diseases on let's say a dozen samples, just because the changes in the composition of the tissue can be very substantial. That analysis does have some issues, and so what we do is actually shift to a simplex space, and then this is a projection of first two principal components in that simplex space. A space that defines essentially the composition of the tissue. And what we're showing here with different colors, so the tumor and benign samples, and you can see that they separate, and the arrows show the loadings on the first two principal components. And here, for instance, these B cells that separate benign samples, and obviously tumor cells and other things separate the tumor samples. So in that space, we could do better statistics. Now, let me give you an example. This is an artificial example. So what we do is we have unperturbed sample shown in green here and perturbed sample in red. And what we do is artificially increase the proportion of cell type 1 and decrease proportion of cell type 2. So if we did this in proportions, then what you see basically is that proportions of all the other cell types are actually affected. You start seeing differences, let's say in cell type 3, even though we didn't really change cell type 3, but our basis has changed. So what we're using is this compositional data analysis technique called CODA, and it basically runs statistical tests to determine in a more robust way what has changed in the tissue. Similarly, you could look at this as sort of a tree where the first change involves both cell type 1 and cell type 2, and then other, let's say, cell types, 3 and so on, remain unchanged. So it's basically a more robust way of analyzing proportions. This is the result for this bone marrow study. This is, for instance, difference in these B cells that I already pointed out. The tumor is much higher in the tumor fraction as you'd expect, as well as macrophage populations, myeloid populations in general. This can be applied to a variety of diseases. In fact, we're doing this. So here's a very quick example from schizophrenia. Again, similar design. In this case, we're extracting nuclei in collaboration with Konstantin Khodasevich. And this is an annotation of the different cell types we see in these cortex samples. Here, again, plotting just control and schizophrenia cells with different colors, and you start seeing some kind of gradual differences, but it's hard to work out. And this is the result of this compositional analysis. In this case, the result is actually fairly simple, surprisingly so. Here I labeled all the inhibitory neurons types in blue and excitatory in red, and you could see higher proportions of excitatory neurons in schizophrenia samples for some reason. Now, this analysis that I showed you depends on the formulation of these cell types, right? And that's not always straightforward to do. You define more granular types, more coarse types, let's say group all excitatory neurons together if you want, or go in and do really detailed analysis. So to some extent, we want to make sure that we have alternatives of, let's say, compositional difference analysis that don't depend on this resolution of clustering. And here's an example. We basically use density, cell density in some kind of space. In this case, the density is calculated in this embedding space. You could also calculate it in this kind of a joint graph space. And we have control samples, schizophrenia samples. The density is calculated for every sample separately, normalized, and then added up. And once we have this, we basically have data for every point here. We have density estimates from every sample. So we can start doing statistics on this. So here we show z-scores, and high z-score, the yellow one, indicates that this population tends to be higher in schizophrenia with some statistical significance. Now, the thing to point out that some of the populations, let's say this a population of SST neurons, they're fairly uniform in terms of their behavior, so they're higher in control. Whereas other clusters like this very large L2-CUX2-LAM5 population actually behaves differently. So some parts of it are high in schizophrenia, some parts are high in control. And these types of cluster-free analysis allow you to check basically whether the granularity of your populations that you've defined accurately captures the behaviors that you see in the data. The other thing I wanted to describe is sort of the flip side of the compositional differences, the transcriptional differences. So you can have cell type that's present in the same abundance in both, let's say, disease and healthy state, in healthy samples, but it changes its state, right? So it changes its expression pattern. And obviously that can be very important for figuring out the mechanisms underlying the disease. So here, first, we're just going to calculate for, let's say, a pair of samples, how much did each cell type change? So we can measure some kind of expression distance between two samples for the same subpopulation. And we can do this for all the subpopulations. And the first thing we'll do is just look at this kind of weighted average, right? So it tries to estimate, the weight is according to the size of the population. So it tries to estimate the total expression differences that we see between two samples. So if we do this, we have basically distances between all the samples, right? We can embed them also into 2D space, in this case using t-SNE. So every point here is a sample, and then the colors and the shape, the colors represent different patients and the shapes represent different types. Now, you can see a few things. So all crosses, red crosses are all controls, right? So the controls cluster together, and then the cancer samples are sort of spread out around them. And the other thing you could see is that you often have a triangle and a circle that are right next to each other. And that's because this is involved and non-involved bone marrow, right? So they're very simple, similar types of samples from the same patient. And realistically, because the patient has its own expression patterns, right? These samples end up close together. Now, we could quantify this overall behavior. So here we're looking on the y-axis, the expression difference between patients, right? So every point is a pair of patients, let's say in this case, benign controls, in this case, non-involved, involved, and tumor samples themselves. And what we see is that as we sort of move towards the site of metastasis, the interpatient expression distance tends to increase, meaning that, when we get to, let's say, tumors, the patients in terms of their expression state, they start to look a lot more different than the benign controls did. Now, this is actually a trend that we see across a number of diseases. So here, going back to schizophrenia, very similar picture. The controls are much more similar to each other than the schizophrenic patients. Now, this just tells you about the distance, right? And one way, to look at that distance matrix is using some kind of an embedding. So here we use multidimensional scaling to basically project these samples into 2D space again. And what we see is that, again, the controls sort of cluster together, but the schizophrenic samples, they tend to, not go in all kinds of different directions. Now, that could have been different. They could have formed, let's say, two clusters, right? That would suggest that there might be subtypes of disease that could be followed up. But in this case, it seems to be spread out. One way or the other, these types of overviews of distances, between samples are very useful for screening the structure of the population. And here we do this using this kind of weighted total distance across all the cell types, but you could obviously do this for every individual cell type, with respect to, let's say, NK cells or myeloid cells and so on. Okay, so the transcriptional shifts, right, can also be estimated for every individual cell type, and the question they're trying to answer quite often is that what cells are impacted the most by the disease? Because if you don't have sort of a preconceived notion, you may want to start by analyzing and following up on the most drastic changes that happen in the tissue. And so this type of ranking of cell types by the distance, the disease versus healthy distance, it becomes quite important. Now, measuring distances in high dimensional space, like between 5,000 transcripts on one side, 5,000 on the other side, is difficult. You have to reduce dimensions. You have to worry about all sorts of covariates. So what we ended up doing is actually normalizing each cell type on its own, because cell types also differ systematically in terms of their transcriptional properties, technical properties. Some have basically more mRNA molecules, and you end up having deeper coverage of these cells. So what's shown here is a standardized distance. So it's a distance between conditions, between, let's say, tumor and controls in this case, divided by the distance within conditions. So the distance is, let's say, between controls. And that gives you some kind of prioritization of cell types, in this case, progenitor cells, for instance, or PDC cells most changed. Again, you could have a cluster-free version of this, of expression shifts, to try to prioritize the areas of embedding, in this case, where the difference between disease and control appears to be most striking. And that can give you some sort of prioritization, again, on what to follow up. In the end, you have to do differential expression analysis if you're trying to interpret what is actually the nature of that expression shift, what changed between disease and control for a given cell type. And here you have a bunch of control, let's say, bunch of cells in control samples, bunch of cells in disease samples. And the method that's being used currently, and it seems to be the best one, is a pseudo-bulking method. So basically take all the cells that we measured from a given sample, and we collapse them into essentially a bulk RNA-seq profile, right? So forget about molecular barcodes. And then your problem becomes very familiar. You have sets of bulk RNA-seq samples, right, between disease and control. And you can use familiar tools such as DSeq2 or JAR to actually run the differential expression. And that's what we use. However, it doesn't work so well in many cases. So there are limitations and one should be aware of. And the main thing is that the actual stability of these results is not great. If you have a cohort, let's say, of 10 patients and 10 controls, right, that's a sizable cohort being measured. And you could do leave one out experiments, right? So we're going to take, let's say, one of the control samples and one of the disease samples, and then rerun differential expression. And then look at stability of the results for the top 100 most differentially expressed genes. And it turns out that stability is actually quite moderate, right, or poor. 0.5 Jaccard index is not great. If you do a p-value cutoff, it becomes abysmal, right? So it doesn't work well. And we fought this for a while. It doesn't depend on the cutoff so much, or p-value or top-end genes cutoff. Improves a little bit if you get more genes. But it seems to be basically the general property of these types of tests. So here we do random group assignment, right? So this is a null hypothesis testing. And what we see essentially is that the distribution of p-values is not uniform, right? And that's supposed to be the case for a test, right? Any statistical test under null hypothesis. And we see inflation of p-values basically at the most important tail, right? Very low p-values. So this probably has to do with the assumptions that are being made by the tests, but just something to be aware of. We don't have a fix for it. Obviously, you could do overviews of what's happening. Pathway analysis is very useful. So here, for every cell type, we run either GSCA or just pathway enrichment analysis for top-end genes. And then we produce this type of summary heat map. So that means that across all these cell types and K, MDC, and so on, you had this ER pathway, which is basically protein folding response pathway, for instance, being significantly enriched in the upregulated genes, and genes that are upregulated and involved bone marrow right next to the site of metastasis relative to controls. Same thing for downregulation. There's nothing fancy about this. It's just a convenient way of getting an overview as to what the functional changes are in the tissue. All right, so I'll just very quickly show a couple of vignettes about this particular study. So here, again, comparing the tumor samples and benign samples, we see very large differences in these myeloid populations called TIM and TAM. So these are macrophages, these are inflammatory monocytes. And then on the T cell side, right, so this is just detailed clustering of T cells. What you see is increase in these types of CTL2 populations in particular, and those tend to be exhausted subpopulations, so exhausted T cells, so they have increased expression of exhaustion markers and decreased cytotoxicity, which is a classic pattern. And so in this particular study, we thought that we could try to link the two, the appearance of these myeloid subpopulations, such as repressive macrophages, and the decrease in effectiveness of these T cells that are supposed to attack the tumor. To some extent, you could correlate them by looking across patients. So here we're looking at abundance of these repressive macrophages per tumor. And on the y-axis, we're looking at the level of exhaustion in the CTL2 population, and they do correlate, right? So that's a useful screen, especially if you have more samples. And in this case, you know, we looked at sort of potential signaling axes that could mediate this type of communication, and ended up picking a particular one. So CCL20, CCR6 seemed to be most promising, both by analysis and by the literature that was available. And so what followed then is a set of experiments to try to validate that particular axis, whether it had an impact on the T cells, their exhaustion state, and the survival overall. So we designed with our collaborators a mouse model of bone marrow metastasis in particular, a cell line that localized to the bone marrow, and then followed up with a couple of perturbations. So one was to knock out the receptor and monitor tumor growth and survival. So if we do this by simply knocking out CCR6, we see improved survival in the knockout. And the reciprocal change is to use CCL20 blocking antibody to basically interrupt this type of signaling. And again, we see improved survival in the animals when this signal is blocked. Yeah, so just a quick summary. I wanted to kind of illustrate comparative study designs using single cell data. It can be more than two groups, but you know, for most of statistical analysis, we're really focusing on one, let's say, cases versus controls. There are two kind of flip axes there. One is compositional shifts, which cell types increase or decrease in abundance. And here, you know, you have to worry about compositional analysis and effects somewhat. And the other axis is transcriptional impacts, so which cell types have changed the most. In all of these statistical tests, it's important to remember that the key difference we're trying to overcome is inter-individual variation. This is where most of the noise comes from. And when you're running a test like, let's say, differential expression test, you're mostly trying to see whether the difference overcomes this inter-individual variation. It's also important to remember that there can be all sorts of covariates and confounding factors in the design. So when you have a two-group design, you have to worry about, let's say, age of participants, you know, the conditions in which the samples were treated, and so on. Otherwise, it will confound your analysis. Okay, and with this, I'll just want to acknowledge my collaborators. So this was a pretty big team for the bone marrow metastasis project with hematologists, oncologists, surgeons, and a lot of postdocs. And I also wanted to acknowledge my own lab. The preprint of this bone marrow metastasis work is on bioRxiv. Thank you very much.
As far as I understood, that you have analyzed some expression level, so mutation rates or so on, but did you try to use gene ontology or molecular pathways? Generally, in terms of molecular pathways, if data are aggregated, the differences between healthy and disease states, between populations, between ages are more articulated than then if we took only genes, distinct genes?
Absolutely. So I kind of skipped through that screen pretty quickly, but the heat maps that I showed, the red and the blue heat maps, these were pathway analysis. And you're absolutely correct that because the data is very noisy, grouping things and testing for pathways, of course, gives you better statistical power. I should note that I talked about this problem with stability in the DE analysis currently for these relatively modest collections. So that problem actually exists for pathway analysis as well. So even though it gives us sort of better statistical power and better interpretation, of course, it does not solve the sort of bias issue that we see in the DE analysis.
But did you try some machine learning methods with blind verification, blind validation?
For DE, for differential expression?
For disease types and differential expression, for example.
Yeah, so for differential expression, most of the methods are based on kind of statistical models. So let's say negative binomial model. They fit parameters of that those distributions.
But we have some more sophisticated machine learning methods. So sometimes they are quite powerful to recognize even very slender differences, such as prognostic power for different drugs and treatment types and treatment regimens.
Yeah, I think there's space for development here. So, what we're pointing out is a limitation of the current, these popular methods anyway. And so I think going forward, it's worth looking at other alternatives.
But I'm a strong advocate for pathway analysis combined with sophisticated machine learning methods, because current bioinformatics is overflooded with some kind of dull differential analysis and p-value mining.
That could very well be. I mean, at this point, we're just trying to figure out what the sort of recommendation for general approach is. And I agree with you that that there are probably more sophisticated tools that could be tested here. But this stability criteria is, I think, one thing to keep an eye on.
Okay, thank
you. Peter, can I ask a very short question? Since we are projecting for, say, Russian audience at most, so a lot of people have interest of involving single-cell RNA-seq in the studies, and there is kind of very wide variety of opinions. So some people say, okay, you can use one control and one sample because so many cells inside, so they can be kind of normalizing factor. But just for you, what an advice from your side?
Yeah.
Unless you can afford, say, 5 and 5 or 10 and 10 and 2 and 2, don't even think about starting because it's a waste of money. Something like this. So even short advice would be very much
so short advice. There are different purposes, right? So if you're just trying to map the composition of the tissue, you can usually get away with just 2 replicates to make sure things work, right? However, if you're doing comparative analysis, all depends on how much variation you see between your samples. So if you're working on mice, they're isogenic, they all look the same, they're all in control conditions, you don't actually need a lot of replicates, 2, 3, and you'll be fine. But if you're looking at humans, they're genetically diverse, they have different history, the environment's different, then you have to overcome that inter-individual variation. So, a few samples not going to cut it. You need, a dozen samples on each side or something close to that.
Thank you, Peter. Thank you very much.